the past paper that I was solving. There are some questions that I want to understand.
Okay, so let's start with the CP10. So A level, so investigate the effect of light intensity, light wavelength, temperature, and availability of the carbon dioxide on the rate of the photosynthesis using the suitable aquatic plant. Right. Uh, Pondweed means algae, beaker, sodium hydrogen carbonate, aluminum foil, spatula, light filter 40 centimeter, paper clip, bench, method. Place a piece of pondweed beaker of the water, cover the with aluminum foil, block the light, cover the other side of the beaker with the help of light filter, add the half spatula of sodium hydrogen carbonate of the water, provide the carbon dioxide, leave the 5 minutes, place the beach lamp a set distance of the beaker, set a photo sensitometer, leave it 5 minutes, record the volume of gas produced during this time, replace the filter with the another color. Okay. What are the hazard? Means what the things we need to avoid? Gas produce. Hmm. So what is that? There is nothing. Like it's very simple. Actually, there are three. What the task is? What is the objective? Right. Objective is we have to investigate the effect of light intensity, light wavelength, and temperature, and availability of the carbon dioxide on the rate of photosynthesis using suitable aquatic plant. We have to use the aquatic plant, and we have to investigate the impact of light on the rate of photosynthesis, right? And there are various factors like temperature, carbon dioxide, because we have studied in detail that these are the factors which affect the carbon dioxide. So here the experiment is saying that you have to prove the effect of different factor including light on the rate of photosynthesis, right? These are the equipment which we have to take. Then there is a method, right? What is the way to do the experiment? How you will do the experiment? Uh, Place a piece of pondweed in a beaker of water. You have a beaker. You have to put algae on it. Any pondweed which is found in the pond, right? That may be any aquatic plant. Cover one side of the beaker with the aluminum foil. So block out the light. First, we have to cover the beaker with the aluminum foil so that light do not go inside. Cover the other side of the beaker with one of the light filter. We have to use the light filter so that light do not go. A half of a spectra of sodium hydrogen carbonate water provide the carbon dioxide, right? To provide carbon dioxide, we have to, because you have blocked the beaker, so carbon dioxide from the external environment will not go inside. So when you will drop the sodium hydrogen carbonate, right? Sodium hydrogen carbonate, what is that?
A powder?
What kind of water? What is the name of that powder?
I don't know.
That is baking soda. Come on, NaHCO3. You can call it sodium
NaHCO3?
NaHCO3. Yes.
Oh, okay.
So what is that? That is a baking soda, and it always react with the water and it produces the carbon dioxide gas, right? Either you call it sodium hydrogen carbonate, or you can call it sodium bicarbonate, or you call it baking soda. That is a common name. Leave it for the 5 minutes. When you will drop this sodium hydrogen carbonate, what will happen? The carbon dioxide production will take place. So you do not, the plant do not require the carbon dioxide from the external environment, because you have already blocked the external entry of the gas. Set up the photosynthetometer. Photosynthetometer is a device, right? And that is a digital device, right? It tell you how much, like what is the photosynthesis rate of the photosynthesis, right? Nowadays people are using it for agricultural experiment. Record the volume of gas produced during this time, right? You have to record what is the, how much the gas is produced, right? Carbon dioxide. Replace the filter with another color of the filter and repeat the experiment. So first you have to use the filter, then you have to use another semi-transparent thing, then transparent thing. So and the different light intensity, what is the rate of photosynthesis in different light intensity, right? What can be
Sir,
the, yes,
I wanted to ask something.
yeah.
How can we record the volume of gas produced?
There should be some equipment for the gas production rate, but here here we have to just tell the rate of photosynthesis.
Okay.
But there are some there are how you will come to know what is the intensity of light. So for this experiment, first thing you require the photosynthetometer, that is a digital thing that comes. Second thing, luminous meter, that will define that what is the light intensity in the lab.
Sir, when we place the bench lamp, should we make sure the bench lamp is facing the aluminum foil or the other side?
Other side, not the aluminum foil.
Okay.
Biohazard, what can be, means the harmful kind of effect. Risk, what is risk? Contamination. What kind of the contamination? Use disinfectant, wash the hand with the soap after handling, right? You may get infected from the algae, any bacterial infection from the water and all that. Seeking assistance, and risk management is low. So we always do the experiment when risk management is low or medium. Lamp, temporarily damage the eyes if directly the light is falling on eyes, that for the sake of some fraction of time we can become uh, uh, blank, right? So that is possible. Wait after image to disappear, see appropriate assistance needed, right? Risk management is low. Sodium hydrogen carbonate, low hazard, avoid the inhalation, contact with the eyes, right? Wear the eye protection, avoid the contact with the skin. Seek medical assistance, seek assistance when if you think that there is some problem. Plot a graph of the color after filter against the volume of the gas production. Okay, so we have to plot a graph, bar graph.
Sir, what will the graph look like? Where should I write volume of gas? On the
Look,
y-axis or
if
x-axis?
the rate of photosynthesis will be more, right, then the carbon dioxide production will be more or less. Sorry, oxygen production will be more or less.
More?
More, right? If intensity is less, so what we have to do, you can do one thing. You can take the light intensity on the x-axis, and because you have to make the bar, and you can take the gas production on the y-axis. Got it? Look,
But sir, it says color of filter against volume of gas.
look. Volume of gas produced, right? Here we can take color of filter, right?
Okay.
Suppose that this is the red color, right? So in red color, the rate of photosynthesis will be more or less?
More.
More, definitely more. So gas production will be more. Now come to the blue color. In blue color, should it be more than red or less than red?
Less than red.
No, more than red.
Okay.
Just a minute, hold on. The maximum photosynthesis takes place in mixture of red and blue. Yes, you are right. It should be less than. The rate of photosynthesis will be low in blue light. So a bit low, not too much. And right, blue. Then we can see that yellow, take yellow color, it will be less, right? Green color, it will be least, right? Almost no. So how you can plot the graph? This is the bar graph, right? If say they say you have to draw the chart, you can chart. Look, draw it like this way. Okay. Here, what is the amount of the gas produced? So amount of the gas can be represented in cubic, right? So that's how the graph plot will come. Got it or not?
Yes. So
So
like the color filter.
color
Okay.
filter will define the intensity of light. That's it.
Yeah. Okay.
Yeah, that is the same thing.
Sir,
Here
if I'm asked to tell the conclusion of this graph, what should I write?
you can write the maximum photosynthesis takes place in the red color, then in the blue color, and then another color, and put one. You can take one more thing, white color without any filter. If there is a white light, right, you can conclude that the maximum photosynthesis will take place in the bright light, white light, then in the red color, then in the blue color, and lead photosynthesis can takes place or no photosynthesis takes place in the gas, the green color, because
Green.
when the green color we drop the green color, the gas production is the least, and at the white light the gas production is the maximum. That's it.
Okay. Sir, if
And
I'm asked why does red light cause more photosynthesis, what should be my answer?
your answer should be because the red wavelength is the maximum. So when the red light is, we drop the red light, uh, uh, both photo system 1 and photo system 2 both are the working. Sorry.
Okay.
In the wavelength of the red light is 700 nanometer. So 700 PS7, PS680 and PS7. Yes, both of the photo system will work in the red light, presence of red light and white light, right? That's how the way light that's how the photosynthesis depend on the basis of wavelength.
Sir, why without any filter? Why does the
When you will
photosynthesis go?
when you will not when you will not use any filter, all kind of the color, all 7 color will go, and on that time all antenna pigment will work, that will capture the light, all photo system will work, so the maximum photosynthesis will take place. When you will put the filter, many of the antenna pigment they won't be able to capture the light, because the light which they capture, that is not present in the spectrum. Got it? Got it or not?
Yes.
Because there are certain antenna pigment which capture, there are some capture yellow light, some capture green light, so some capture the violet light, right? So if those lights are not present, then those antenna pigment won't be able to capture the light, and if they won't be able to capture the light, the rate of photosynthesis will be low.
Okay, so all antenna pigments absorb light in
In
filter.
white light, because in white light all
In
frequencies are
white
present.
light. Okay, so no filter is white light, right?
No filter is a white light. The sunlight is called white light, though it appears yellow to us, but it is not yellow. Actually, this is the white light. Why it appears the
Okay.
yellow to us? Because the yellow color is scatter least in the dust. Got it
Okay.
on answer or not?
Yes.
Sunlight is actually white light, but it appears yellow to us because yellow is scatter more least. Is that clear?
Okay. Yes, sir.
That was the experiment. Nothing else. What about the next one? Just a minute. Carry out the study of ecology of a habitat, such as using quadrat and transect, determine distribution of abundance of the organism and measuring the abiotic factor appropriate to this factor. Carry out the study of ecology of a habitat, such as using quadrat and transect. Okay. Determine the distribution and abundance of organism and measuring abiotic factor appropriate to this factor. Various abiotic factors that affect the abundance of the distribution of certain species can investigate this ecological study. Sub example: light intensity, distance from large stream, river depth, equipment 0.25 meter quadrant grid, 20-meter belt transit, clipboard and pen identification book, measuring the equipment depending on the factor. When carry out the ecological study of a habitat, there are two main methods of sampling: transect and quadrant. Method 1: quadrant. Draw a large grid over the image. Okay. For the map of 2/3 area being sampled at. Assign a numerical scale of grid, creating a coordinate grid. Use the random number to generate 20 coordinate first area. These all will be quadrant are placed. Place a quadrant at generated coordinate and count the number of the species being investigated in quadrant or percentage score. The box of the quadrant more than half filled by the species being invested. Count the full box and calculate the relative base step 3. Look, what you have to do, you have to do actually here two experiments. You have to take a habitat, right? And we have to use the two method, quadrant and transect, right? So what is the quadrant method? Do you remember quadrant method or not, Affaf? Quadrant method, do you remember?
Yes, sir, roughly.
Roughly. Okay. So quadrant, the quadrant should be of 0.25 meter square. Means one arm should be 0.5, right?
Okay.
Means half of meter, right? 0.5 means 50 centimeter by 50 centimeter. That should be the arm of the quadrant, what we are taking. And 20 meter, we have to take the area of 20-meter belt, right? So, and here we have to draw the quadrant. Then we have to take the pen and all that, right? So in the quadrant, what we have to do, you have to approximately, we have to take the, or we have to map the 2/3 area, right? Sample, right? It's a very big task. And now, suppose that we have taken the 20 quadrant, right? For sampling. In 2/3 area, we have taken the 20 quadrant. In all the quadrant, we in every quadrant we have to suppose that I have chosen 5 species, right? So we have to take the number of species per quadrant. So every species need to be noted down, right? And they have showed that if almost all the part, 75% part is covered, right, under the quadrant, then only will include this species, or we won't include this species, right? This is completely covered in quadrant, this is 75% covered, this is almost 70% covered. So that's how we will count. Those here only 50% covered, only here 20 or 25% covered. So these will not be counted, right? That we have to take care of. After the sampling, what we have to do, second is transect method. In transect method, suppose that we have to take the those for instance, under the tree, those species like distribution under the tree shaded area and out of the open area, so non-shaded area. So again we have to draw the quadrant. In quadrant we have to take two kind of the habitat, the habitat which is under the shaded area, which are under not which are under the open area, right? Then at every quadrant we have to count the species. Then this is all risk management. What can be cut infection, allergy, there may be allergy, there may we may get infection, we may sleep, leeches may bite us, sunburn can take place if it's a sunny day. Then in first quadrant, so in grassland, we have to take the sample of particular species, sample of second species, third species, that kind. It may go up to 20, right? Uh, Percentage of the frequency, what is the frequency of those plant which are found in particular quadrant. Then in woodland
Okay,
means we are,
sir.
yes,
I wanted to ask, in the grassland, we have to sample different kinds of plants,
different kind,
like
yes,
the same
different
plants
kind of
in
species,
different,
different kind of
different kind of
species.
species. Okay,
Yes, that is the
okay,
only ecological diversity. What is ecological diversity? The number of the species of plant or animal present in particular area is called frequency.
okay. Sir, how can I calculate percentage cover?
Simply, suppose that we have a quadrant of 50 by 50, right? 50 centimeter wide, 50 centimeter longer. In that, there are 10 plants, right? So, what should be the frequency? What is the frequency percentage? Right? Suppose that those 10 plants are covering the 50% of that quadrant. You can route roughly the 50% frequency.
Okay,
The same thing will go with the plant, tree like plant, small tree like, right? Again, the sampling we have to take. Now, here, the light intensity, other abiotic factor, the frequency, the cover or frequency, light intensity or area 1, area 2, area 3. Okay. distance among transect 0, 2, 4, 6. What is that? Okay, transect method. Sorry, this is the second method. In area 1, where the light intensity, you can write it down. Light intensity is low, right? Low light intensity. The area, the frequency, the covered means the species. Here we have to write down the species. Light intensity, suppose that in second area, the light intensity is full light intensity. The frequency, here we have to route. That's how we have to cover the three areas, right? You may write here that partially covered, partially open means optimum light intensity. Then the species cover. Now here what we have to do, we have to use some statistical tools, right? Two tools, Sperman's rank correlation coefficient. Here we have to put in the formula, and quadratic mean percentage cover frequency. T-test, we have to use the t-test. Here we have to use the two formula. By the way, I don't think that you are going to do this experiment on field.
No, we are actually going to write about it.
Exactly.
So
You
like,
have, yeah, better to take the data from somewhere, from some previous year questions, right? Or some book where you get the, you must have some practical book at your like any bookshop and all that.
okay.
So
So
take,
actually, it's going to be more like they were written a question that, oh, this person went there and then he measured with the belt transect this much. What can be the conclusion? So
so
they
yes,
write question like that.
take some example and learn the example, right? So that's how you have to do. Otherwise, it's a very clumsy kind of experiment,
Okay.
because it's very difficult to do the experiment. Suppose that if somebody gives this experiment, I will take 15 to 20 days to complete it. We used to do this experiment on in master's classes, master MSc botany and all that. And even and the result used to be very. So better take some example, put some value here and there, right? Suppose that you got the example from any book, right? You have to play with smartly. You have to play with the value, right? And then write it down, and that's just copy and paste the same thing over here. That is the only possible thing which you can do. And in fact, that all the student will do. Who will cover this or will attempt this one. So all the student they have to do. You need to learn only the methodology, because will they take the viva or it's a written exam only?
Only written exam, sir.
Oh, come on. So then you have to do one thing. Just cram any example and just copy and paste over here.
Okay.
Get the example from any practical manual kind of the book that will be available in the market. Take that book, cram some data for this one and even the first one. Cram some data and copy and paste. That's it. And that is the way to take the practical. What is this? This one? What is this
This is
one?
actually a question. I know the answers to these two, but there's the third part that I want to know the answer of.
There is a third question.
Yeah, this one.
This one.
Yeah.
This is the coleoptile experiment which done by the Darwin, right? And this experiment just try to prove that the auxin is responsible for phototropic movement, right? Because of the phototropic move, because of the auxin hormone. Remember phytohormone auxin? Because
Yeah.
of the hormone, plant move in the direction of the light. This is the small seedling, right? And this is the coleoptile. It's a kind of the grass, right? Coleoptile, right? When you will put it to the light, this it will move toward the direction of the light, right? When tip is present, slice of the tip, because auxin is found in the tip. Try to understand. Auxin is found in the tip, right? If you will remove the tip, means you will remove the oxygen, then plant won't respond to the light because oxygen is not present over.
Okay.
Then agar block is placed on the here you place the agar block externally, agar block. Then you will put the light, but it is not responding because oxygen is not present there. Then what you are doing, you have taken the complete coleoptile, but you have put a plastic or some sheet over here, right? What actually oxygen do? Let me tell you what actually oxygen do. When light is coming from the front side, right? The light is coming from this direction. Oxygen will accumulate here, opposite to the direct light direction, right? And it will enlarge the cell. It will increase the size of the cell. So if the size of the cell will increase, the coleoptile will move in this direction, right? Got it?
Yeah. Yes.
Here you have removed oxygen. Oxygen is not present. Here oxygen is not present. Here oxygen is present, but when you will drop the light, you have put this plate. Oxygen will not go downward, accumulate here. So the cell enlargement will not take place. When you will put the sheet in this side, front side, what will happen? Oxygen will be able to diffuse over here and movement will take place. Right? Experiment this. A gel layer is placed below the tip of the coleoptile and coleoptile illuminated. You have placed the gel, but oxygen will diffuse it, and so because of the diffusion of the oxygen, it will bend towards the light. What is it?
Sir, even though there is gel layer, why does it still move towards
Because
light?
gel is the thing through which the chemical can diffuse.
Okay. Okay.
But here this is because gel is the permeable thing, right? Gel is the permeable kind of the thing, but this is the impermeable. This layer is impermeable, so the oxygen will not diffuse.
Okay. There's
So this
a third
experiment
experiment.
tried to prove that oxygen is found at the tip of the plant, and when light falls on the coleoptile tip, oxygen goes away from the light, cell enlargement take place, and the coleoptile bend toward the direction of the light.
Okay. There's a third experiment, sir. Yeah, this.
Again, the same thing. Coleoptile illuminated, removed. You have removed it. It won't show any movement. Replace on the other side, remaining the coleoptile. Then the movement will take place. Why? Because the coleoptile is present, the diffusion will take place.
Okay, so the tip is replaced on one side of the remaining coleoptile that is then kept in the dark. It is
Exactly.
kept in the dark, but it still moves. Why?
Tip replace on the remaining coleoptile, that is coleoptile that is taken. Just a minute. Coleoptile illuminated and the tip is removed. Nothing happened. Tip is replaced on one side of the remaining coleoptile. Okay, first the tip have been illuminated. Tip have been illuminated.
Oxygens
Then
are activated.
exactly, oxygen is present, right? Means it have accumulated opposite side.
Okay.
This tip, because tip is already illuminated, so oxygen
Okay.
will diffuse out. Means oxygen is there, activated. Clear?
Yes. So like this is the question.
This
Can you
experiment
scroll
directly,
down?
actually, this is not the new experiment. This experiment have been done by the Darwin, and that's how the oxygen was discovered. Then this
I
one,
know this, sir. I know this. No, this is just related to the question I wanted to ask. Yeah, this is the question I want to understand.
this one,
Yeah. Just scroll
uh,
a bit down.
speed of conduction meter per second, neuron diameter. In non-myelinated, very simple. I have already told you that myelinated neuron, the myelin sheath is responsible for the increasing the speed of the nerve transaction multifold, right? neuron diameter, that is the diameter of neuron, thickness of neuron, right? As the thickness of neuron is increasing, the speed of conduction is increasing, but gradually. But when it is for the non-myelinated, because in non-myelinated, let me tell you what happens
Saltatory conduction doesn't take place.
exactly. And in myelinated, the charges start jumping from here to there, and like frog, and that's how the nerve conduction takes place. So you can see here the as you increase the diameter, the rate of speed conduction is going faster and faster. That's it.
No, 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 sir. Scroll down. Yeah, this. How do I do this?
Equation y mx plus c. C is the speed of the light. C is the speed of the nerve impulse. The gradient for non-myelated neuron is 0.5. Okay. And then what is y is equal to mx plus c? What is m? No, no, no. C is the speed of the light. It have not been given over here. What is the c? Is it there? What is the c?
No. No, you
No.
have to figure out what value is what.
Y is the value of when y axis. X is the value in the x-axis. This m gradient, m is the gradient, is coefficient 0.5. It is given. What is the value of c? That I don't know. What is the c?
But this one we are doing for non-myelinated neurons because
You can do for both.
no. See, the gradient for non-myelinated neuron is 0.5. The gradient of non-myelinated is already known. So we have to understand the myelinated neuron. So
Okay.
y value is going to be 120
Y value
because
going
that's
to
the
be
highest point of
120.
myelin.
Where is, yes, 120, right?
Yeah, why
Right.
is that? Then x can be 20 because that's where the last point is.
Exactly. M is 0.5. Then value of
And no, I guess,
gradient. This is the gradient.
yeah.
M is the coefficient. Calculate the percentage difference in the gradient. Hmm.
M is the gradient. We have to find that, sir. And then we can find the
Then what is
percentage
0.5?
difference. No, they just, no, this value is given for the percentage difference to find out the percentage difference. Calculate the percentage difference in the gradient of myelinated and non-myelinated neurons. The gradient for non-myelinated neuron is 0.5. So
Okay.
first we have to find the gradient of the myelinated neuron, and then we can find the percentage difference.
Then we have to get the gradient for the this one non-melted. Then we have to get the value of m. Then what is the c? Actually, the question is that what is the c? Is it the speed of the light?
I don't know.
Because c in physics, c represents the speed of the light. What is the c over here? Even I don't know. What is the c?
Yeah, and this question, I wanted to ask that one as well.
Just a minute.
Sir, according to Google, the C is the point in which the line crosses the y-axis.
Just a minute. Let me search. Let's see. That's what I am searching. Uh, MX the equation MX is in general equation is straight where the gradient P is y-intercept, then interception is not taking place over here. I will define the C. I I'm not getting this. How to get there? Because the C is the y-intercept, but here
So
interception is not.
the C can be the part where the line meets on the y-axis. So it meets at 0. So it can be 0.
Like we cannot say like that. Are you taking the maths class as well? From
No,
someone?
sir.
No. Who's in touch with you in physics?
No.
Because physics, even the physics teacher can tell you. But they will tell you in the terms of physics. Yes, but they can solve it. Uh, just is it, I'm just trying to give my last attempt.
So can you move to another question?
Sure. Is there any other question as well?
Yeah. Those are also math related.
Mm. Mm.
Yeah, this one.
Antitrypsin deficiency, inherited disease that can affect the different organ system, protein producing. Investigate the breathing of the patient, resting tidal volume, there is space. Calculate the volume of the air entered to alveoli each hour, AV, so VT, DES, RR. That is simple.
No, but sir, 12 breaths per minute, but we want AV to be each hour.
We want AV to
Each
each
hour.
hour.
Yeah.
Yes.
So R is 12 breaths per minute.
12 breath per minute. Yes. Then each R, then what will be the RR? 12 breath per minute. So per minute, multiply it with 60. You will get the rate per hour rate. Then you will get the AV per hour. That's it.
Wait, sir.
Okay.
Yeah, you're right.
Uh, that is the ECG. Okay.
I know this.
You know.
It's cardiac diastole.
Yeah, yeah. We have
Yeah,
studied.
this one, the last question up there.
This one,
Up.
this one.
Yeah, calculate the mean heart rate shown in the ECG trace.
Mean heart rate. How we'll calculate from this graph. 1 MB. Is
The
there also
volume the volume is, sorry, the value. It's 1 MV is
1
1B. And
1
not
MB?
this one, sir, down. That's a different one. Yeah, this one.
This one 0 second.
This one, yeah.
Both the value are same, 1 MB, 1 MB.
No, but the graph is different. This one is faster, that one is slower.
Yes. And there are 5 cardiac cycle. Here there are 3 cardiac cycles. Calculate the mean heart rate shown in ECG. In 1 second heart rate. Okay, this is 1 cycle. So 1 second, then 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15. So total this cardiac cycle is for the 3 second, right?
Yeah.
3 second. So how many heartbeat are there? Beats are there in 3
5.
second?
5.
5, right? So what will be the heartbeat per minute?
I don't
60 by 3.
60, yeah, okay.
60 by 3, right?
Yes.
So 60 by 3. There will be 20. The answer will be 20. So 20 into 5. So 20 into, okay. There, look, there are 5 heartbeat in per 3 second. Okay, that's how we can get. That's how we can get 5 heartbeat in 3 seconds, right? No, sorry. In 3 seconds, 5 heartbeat. In 1 second, 5 on 3 heartbeat. In 60 seconds, 5 into 60 upon 3. Yes, I was right. 20. So here the rate of heart to 5 ja 10. 100 heartbeat per minute. That should be 72. It's a bit higher. Got it, Affaf?
Yes.
Then the same thing we can calculate by for the upper one. For this one. Now again calculate again. Here we can calculate. That is the again the time. I think time should be 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 13, 14, 15, 16. 1, 2, 3, 4, 5, 3, 5, 6, 7, 8, 9, 10, 11, 7, 12, 13, 14, 15. Yes, 15. And 1, 2, 3, 4, 5. Yes. Again here, there are in 3 seconds, just 3 heartbeat. In 1 second, 3 by 3 heartbeat. In 60 seconds, 3 upon 3 into 60. right? The answer will be 60 beat per minute, right? This is the slower rate of the heart. This is the faster pace of the heart. That's how we can calculate the heartbeat. Got it from ECG.
Yes. Okay. Sir, can you ask that question from someone who knows math, the graph one, and then you can
And
discuss it in the next
this upper
class?
one,
Yeah, the graph
I will
one.
send it to Ajam sir. I think most probably he'll tell you because he is a physics faculty. Like he's a teaching physics. Have you encountered with him, Ajam sir?
No,
He is the
I'm
best in
also doing
physics.
biology.
Okay, okay. Ajam sir is taking care of the IT part. I'm taking care of the medical part actually.
Okay.
So yeah, he's my friend and he's the best faculty of the physics as well. In fact, he's the director of Learnivio. So I'll ask Ajam sir this question. Most probably he'll tell me.
Okay,
Okay.
sir, okay, okay, fine.
Yeah, I'll ask him because this is purely mathematical question.
Okay.
Yeah, I'll ask this question to him. Fine. Anything else?